thank the organizers for giving me the opportunity to come over here and present our work, as well as talk a little bit about the company. And also it is very interesting that I believe that this is kind of a beginning of the OMICS group and its effort in spreading the genomics and pharmacogenomics. And I think it is in several years to come, this particular area is going to, I would say not revolutionize, but definitely going to change how the patients are going to be treated. So ultimately, all of us know that in addition to earning the money, we are also some way connected to ultimately understanding the disease process, as well as understand how we can modify using drugs. So ultimately, what we are trying to do is contribute towards treating patients. So actually, my name is Subbu Apparsundaram. I had been the senior scientist of Roche Pharmaceuticals until about 1 year ago. Coming out of Pharmaceuticals, I started this company as a consulting company. We are based in New Jersey at the North Brunswick Bioincubator Center. So while we started operate as a consulting company, we also understood the need of the emphasis on industry, university collaboration, as well as my interest in understanding some of the disease mechanisms that is happening within Indian population, as well as understand the drug effects within specific subpopulations. With that goal, we made a collaboration with Raleigh-Brownstone-University. And this is one of the top 10 universities in India, based in Chennai, one of the major cities in India. And we also have a lab facility in Tysell Park. This organization is actually a science research organization. We also provide some service for the discovery biology, as well as drug discovery work, and some biomarkers research. So I will just briefly go through what we are trying to achieve through this company industry university collaboration. So what our vision is is innovative therapeutics and diagnostics for affordable healthcare. And our mission is actually very oriented to biomarkers. For example, one of the missions that we have is discovery of novel therapeutics guided by appropriate biomarkers. And we also think that it is very important to translate the information from cell-based assays to clinic. And we also focus translational research as one of the discovery research service. In terms of the diagnostic tools, it is driven by biomarkers research, and we apply state-of-the-art techniques. So almost all our research work is being carried out at the Sri Ramachandra University research facility. So what we do? So in terms of, we actually have facility to cover all three areas. Our focus is mainly on translational work, but we also have facility to do preclinical research with animal models, and we also do clinical research through the university hospital setting. We work with small molecules, large molecules, including monoclonal antibodies, as well as biosimilars and biobetters. We have a flex model in the sense that depending on customer need and collaborator need, we can accommodate a different type of research. Because we are in the university facility, we have access to clinicians as well as faculty with different expertise. So we have that kind of flexible model. So in fact, what I would like to emphasize that we are very open for collaborations through the university or through the company, ClinBio. The ClinBio itself is a model where we work with hospitals as well as universities in a collaborative manner. And we also work with pharma biotech companies. So what is our, currently what is going on? What is our main focus? If you ask about that, the two areas of main focus are the pharmacodynamic assays, where we try to understand the mechanism of drug action, as well as we have several functional assays, specific for whether it is a protein-based or a gene-based or cell-based assays. We do many different assays. And we also do a biomarkers research. So basically, as the figure represents here, what we try to do is for any disease model or disease area or drug discovery area, we actually have very good access to clinical samples through our clinics. And using those clinical samples, we try to understand the effects of small molecules or large molecules. And we apply, again, as I mentioned earlier, many of the technologies that is available currently. So in addition to understanding the drug effects, what we are trying to understand also is in terms of the local population, we kind of try to find out, based on the available information on biomarkers, diagnostics that are available in different populations, we kind of validate, actually, is that suitable for the India subpopulation? How can we use these diagnostics? Are these biomarkers representing representing the disease there? Or do we have to find a different biomarker that will be more appropriate for the local population? So we spend a lot of time in the biomarker research. And so what is the goal? Why we do all these things? One of the goals that we have is, as a drug discovery person coming out of Roche, that drug discovery process by itself is a very expensive process. Right now, an average is about $1.8 billion. And also it takes about 13, 14 years. So most of the time when you look at it, the drug discovery process is a very less productive process and a very expensive process. Most of the failures that occur in that process is in the phase 2 and phase 3, where people are looking at lead molecules, the efficacy and safety of lead molecules. So what was happening is most of the test compounds go into the human based on some of the animal and cell-based research, and what people find is at a clinical level it is failing. So most of the cost has gone there. And what our goal is to understand the effect of a compound, both at the level of efficacy as well as the safety, using clinical sample as our model system, so that we can kind of reduce that cost as well as the test compound attrition. So ClinBio Research Service aims to increase success and productivity by providing early readout of efficacy and safety, thereby cutting cost and attrition in drug discovery development. This is one of our primary goals of ClinBio, in addition to understanding the disease process. So without going too much in detail, we focus a lot on the mechanism of action, proof of concept. We use several model systems in terms of assays. These are clinical samples, primary clinical samples, whether it is a human cell-based studies, stem cell-based studies, cancer cell-based studies, neuronal studies, structural studies. So we use, as I mentioned earlier, we focus on early stage pharmacodynamic and functional assays. We work in many different disease areas. The reason we could work again in many different disease areas is that we are collaborating with many clinicians within the university as well as the faculty. And we use many different type of clinical samples for our studies. And we work with many different targets, whether it's a GPCR, enzyme, or cytokine receptors, many different targets. And we use multiple platforms for our pharmacodynamic assays and functional assays. Some of the things we can definitely discuss a lot more in detail. I'm available the next two days or so. So in addition to the small molecule, we are somewhat specialized in biosimilars research. We work with some other companies to develop some antibodies. With regard to those antibodies, we are set up to do potency assay comparisons, the ADCC, CDC assays, neutralizing assays, cytokine storm assays, immunogenicity. So this is almost close to the end of the slides, but so this is, in terms of biomarkers, again, we focus a lot on, we work with many different matrices, and we work with many different matrices, and, Yeah, we work with many different matrices. We apply, beginning to apply many of these techniques. So one of the things that we try to do is profile, understand the disease mechanism, and see whether we can do patient stratification, either for clinical trials or to understand even to drive the drug discovery process. We are actually a one-year-old company. So we are just in the initial stages of many of these technologies. So we currently apply all of these techniques that we talked about to achieve that goal. So why one would think about collaborating with ClinBio, right? That is what the question could be. So one thing we know that we offer many types of collaborations. It's like project collaborations, just for publication purposes we do. In addition to that, we do fee for service, full-time employee basis service. But this is especially that is happening with the pharma biotech industries. We do this a lot. And one thing I have to tell you is through this collaboration, we have very good access to clinical samples. We do oncology research, autoimmune disease research, respiratory disease areas, many different disease areas we work on. And also, we are very particular about ethical. Everything is ethically done in terms of consent forms, very well documented. And the Institution Ethics Committee takes care of all the things that we are trying to do in terms of the research that we do. So ultimately, what we are trying to do is especially find a way to understand the disease mechanism as well as the drug action mechanisms. That's what, thank you. If you have any questions, I can elaborate on any areas.
How do people find out about your company?
Not really, we are not advertising very much, but I'm beginning to do that. Yeah. So we started mainly as a research organization. Sure. Then we are now providing some service also. Only we do mainly rodent, small animals. Mice, rats. So mainly we use for xenograph models for cancer research. That too we are.